Hello, my name is JC Napier, and this is my presentation on oligodendrocytes. This is an oligodendrocyte in the middle of myelination. You may be asking, what are oligodendrocytes? Well, oligodendrocytes are a secretory cell, and are a type of glial cell, which, along with Schwann cells, are tasked with the formation of myelin, and they are normally found in the central nervous system. But what do they do? What are their functions? Well, their main function is the formation of myelin, which, as I said before, which then secreted and then wrapped around axons. Myelin is a combination of lipids and proteins at about a 30 to 50% mix of proteins and 50 to 70% mix of lipids. And in terms of waters and solids, it is about 40% water. An example for a lipid is cholesterol, and the example for a protein is a proteolipid protein. And a very interesting mix that is used is galactocerebroside, which is both a lipid and a glycoprotein. A very important organelle involved in the process of production of myelin is a smooth endoplasmic reticulum. This synthesizes the lipids needed for the myelin and sends these lipids to the plasma membrane through secretory vesicles. Another very important organelle in the process of myelin is ribosomes. These bind to the rough ER and introduce proteins to the rough ER so they can be processed and then sent to the Golgi apparatus. The Golgi apparatus is the third very important organelle in the process of creation of myelin. This receives the proteins from the rough endoplasmic reticulum and then modifies their carbohydrate portions in the glycoproteins. This then sends, once they're done, sends the finished proteins to the plasma membrane through secretory vesicles. The last important part is the plasma membrane. These secretory vesicles fuse with the plasma membrane, and then their contents are released into outside of the cell. Once they're outside of the cell, they mix together to form the myelin. This is then enwrapped around axons to provide insulation.